something. So we have the metabolic adaptation in fasting, in B-oxidation and ketogenesis. So fatty acid in the mitochondria undergoes what's it called B-oxidation. So fatty acid undergoing B-oxidation is the process by which we are breaking down lipids in order to produce energy by glucose infusion. We are breaking down lipids to produce energy. So in the fatty acid, the way by which fatty acid is broken down, the process is called what? B-oxidation, beta oxidation. And during this process, it leads to accumulation of what? Acetyl-CoA. There is a high yield of accumulation of acetyl-CoA. So this excess acetyl-CoA now leads to what? The formation of what? Ketone bodies. And this is why we call this the de novo glucose synthesis. So the word de novo is the process by which we synthesize what we call ketone bodies. So you don't just eat ketone bodies. There is no food that will just give you ketone bodies. What you eat most times are carbohydrates and the rest. Then and lipids. So when you now eat it, the body now convert them towards ketone bodies. So we have three major types of ketone body, which are the acetoacetates, the acetoacetates, the ketone, the 3-hydroxybutyrates, the 3-hydroxybutyrates. And we have three, but we have two here, the acetoacetates and the 3-hydroxybutyrates. So what will now happen? We now take them to the what? To the blood. They diffuse towards the blood. Remember, this is the prolonged state. This is the fasting state. And the fasting state, our body is trying to make energy. Our body is trying to make glucose. This glucose will now make energy we required in our body from non-glucose molecule. So what we are trying to do is we are going to break the fatty acid. We are going to break the triglyceride lipids to produce energy because they yield high yield of energy. And this takes about 12 to 14 hours to occur. The process by which we are breaking down fatty acid or lipid or triacylglycerol is about 14 to 12 hours. So at the end product of this in the mitochondria is acetyl-CoA. So this excess acetyl-CoA is now converted to ketone bodies, which is the acetoacetates and the 3-hydroxybutyrates, which is now diffuse to the blood. Then we now make energy. So when a patient is dying, something must be done. So in the process by which we are converting acetoacetate to the energy our body needs is very, very, very important, because our body does not just convert these ketone bodies to these processes. It will first undergo ketogenesis, and through this ketogenesis is due to accumulation of acetyl-CoA, and this accumulation of acetyl-CoA will now lead to the formation of energy required by the brain whenever glucose have been completely used up, the heart, and the skeletal muscles. So we have two phases. The metabolic rheostat, the insulin and glucagon ratio, the master switch for fuel metabolism. So we are now comparing two constructs for better understanding. So during the first states, what happened? Hyperglycemia, high level of what? Glucose. During the fasting states, what happens? Hypoglycemia, what? Reduce of what? Blood glucose. So let's talk about the fed states before we go back to the low to the fasting state. So what happened? It favors storage. What are you storing? Glycogen. So whenever there's excess glucose, which is called what? Hyperglycemia, it favors storage, by which we are converting those hyperglycemia to what? To glycogen, which is the polysaccharide found in our body. So it also leads to what? Lipogenesis, where they said whenever glucose level is high, we convert them to what? Glycogen to what? Triacylglyceride or lipids. Lipid. And that lipid is what? Fatty acid. We convert this glucose or glycogen to fatty acid. Then from fatty acid, we now combine them with what? Glycerol to form what? lipid. So those processes by which we are forming lipid is like esterification, and this is called what? Lipogenesis. Esterification is adding fatty acid together with what? Glycerol to form what? Lipid, and this process is called what? Lipogenesis. And during the also first state, we also have protein. You might be eating beans, you might be eating egg, milk, and the rest. So we have protein. It will now lead to what? Protein synthesis. So what happens during the fasting state? So in the fasting state, we have it activates AMP cascades. It activates what? AMP cascades. So I'm going to explain something to you now. So we have the AMP, we have the ADP, and we have the ATP. So these are three different molecules. So they are inversely proportional to each other. I'll explain that. So whenever ATP is high, ADP is what? Low, and what? AMP is what? Low. Look at what I mean. Whenever ATP level is high, meaning that we have high level of energy in our body. So since we have high level of energy in our body, we have low level of what? ADP. Remember, ADP is adenosine diphosphate. So when you're missing ADP and ATP, we are adding phosphate to what? ATP. So whenever we have high level of ATP, we have low level of what? ATP. That's why they are inversely directly proportional to each other. So AMP is what? Adenosine monophosphate. It only have one phosphate. So we have low. So what happens during the fasting state? Activation of what? AMP what? Cascades. So it now leads to what? Phosphorylation of what? Key enzyme. It leads to phosphorylation of key enzyme. So flip side towards glycogenolysis. So it will now result to what? Glycogenosis. Whenever there is prolonged fasting, or whenever there is fasting in general, it leads to glycogenosis. And glycogenolysis is a process by which we break down what? Glycogen. We also have what? Lipolysis, breaking down of lipid. Remember that lipid have to yield high amount of energy, but the process is too low. It's too low. Is too slow, taking about 12 to 14 hours. So it's too slow, but during the prolonged fasting it will now occur. We also have gluconeogenesis. We are trying to make energy from non-glucose molecule. Loss of fine turning underlies metabolic disorders. So I remember hyperglycemia, high blood glucose, hypoglycemia, low.